All right, hello everybody. This is part 3 of Respiratory System for Biology 233 in summer of 2018. So when we left off, we were about to discuss the neural control of ventilation, and I mentioned briefly that that phenomenon where you can choose to modulate your respiration, so I can start panting and go if I want to, but I can also breathe while I'm asleep and not actively thinking about my breath. So how might that work? So neural control of ventilation is reliant on repetitive stimuli from the brain. So I'm going to underline repetitive here because, of course, from the time you emerge from your mom and take your first breath, you breathe pretty constantly throughout your entire life, barring any accidents. So the way that works is that your brain is constantly pinging your respiratory muscles to cause you to inflate your lungs. So those neurons occur in populations in the medulla oblongata and the pons. And those are responsible for controlling unconscious breathing. So the breathing that you're doing when you're relaxing, not thinking about breathing, or asleep. Voluntary control is provided by the motor cortex. So you have innervation to the muscles of inspiration and expiration from both parts. So inspiratory neurons are those which act on the muscles of inspiration. So that's going to be your external intercostals and your diaphragm. And expiratory neurons fire during forced expiration. Because remember, passive expiration really just relies on you not contracting your diaphragm anymore and letting it elastically recoil to its original position, as well as letting your lungs elastically recoil to their normal size. Remember those elastic fibers in the submucosa. So the fibers travel down the spinal cord to lower motor neurons, and fibers of the phrenic nerve go to the diaphragm, and intercostal nerves go to the intercostal muscles. So the intercostal nerves are what they sound like. They're between ribs. So they run along the inside of your thorax between adjacent ribs, and those innervate your intercostals. All right, I'm going to pick a different color because I don't like this red. How about blue? Okay, so note on this image, we've got the pneumotaxic area and the apneustic area, and these are involved in breathing as well as the inspiratory and expiratory. So the pneumotaxic and apneustic areas are part of the pons, and the inspiratory and expiratory groups are in the medulla oblongata. So for this reason, as well as others, you really don't want a brainstem injury. It'll get in the way of your ability to breathe, either consciously or unconsciously. So let's talk about what this stuff does in particular. So the inspiratory center is also called the dorsal respiratory group, and that's just relative to the nucleus's position within the medulla oblongata. So these ones control depth and rate of breath. So the more frequently they fire, so more bursts of activity per unit time, the more deeply you inhale. And I'll draw this on the board in lecture, but you can compare the firing patterns of the neuron to predict what breath will look like. Longer duration of firing, so if you get one long burst, that means that an individual breath is prolonged, slowing your average breath rate. The expiratory center, which is also called the ventral respiratory group, is involved in forced expirations. Remember, forced expiration involves screaming, yelling, or involuntary forced expiratory behaviors like coughing. In the pons, you have the pneumotaxic and the apneustic center. So pneumotaxic sends continual inhibitory impulses to the inspiratory center. So let's draw an arrow going up here. So these populations of neurons aren't independent, they talk to each other. And as the impulse frequency rises, your breath rate becomes faster and shallower. The apneustic center prolongs inspiration, so you breathe slower and deeper. All right, so medullary rhythmicity area. This is what it sounds like. So it's in the medulla oblongata, and it's going to control the rhythm of breathing. So anything that has a rhythm has a repetitive pattern that happens over and over again. So the basic respiratory rhythm the one that you would expect to see when you're not having the respiratory rhythm acted upon by sympathetic innervation or emotion, so that you'd see this kind of in coma patients or people that were asleep. So inspiration for 2 seconds, expiration for 3. And this is due to the activity of the autorhythmic cells and then their subsequent inactivity. And the expiratory neurons are inactive during most quiet breathing because, again, you don't need them if you're not forcing air out. They're only active during high ventilation rates, when you need to push air out because you're trying to breathe in again soon, so you got to get that air out faster. So I'm not going to go through these charts in detail because I assume you can read them. It's just showing you which areas act on which things and what happens as a result of their activity. All right, so afferent connections to the brainstem. So this means afferent to the brainstem, particularly not to the CNS. So you get input from the limbic system and from the hypothalamus, our 2 friends in memory and emotion. And this changes the rhythm of your breathing in response to pain and emotion. So things like crying, which is arguably an altered respiratory rhythm, and also panting when you're in pain or other kinds of breathing. You also have input from chemoreceptors. So these are detecting various chemical components in your blood and altering your breath rate accordingly. So we'll get a lot more into the influence of this system when we talk about when we talk about the end of the respiratory system. But for now, just know that they detect things like blood pH. So that's how many protons. These square brackets mean concentration. So this is concentration of blood protons, carbon dioxide, and oxygen levels in the blood as well. You can also respond to inhaled irritants. So let's say you inhale some smoke or some noxious chemical. That's going to stimulate vagal innervation to the medulla, which results in bronchoconstriction and or coughing. So let's add an and here because you can do both at the same time. That absolutely would happen if you were, say, trying to escape a burning building and there was a lot of smoke. Inflation reflex, excessive inflation of the lungs to the point where they're going to be stretched to the extent of tissue damage, stops inspiration. So that's why there's a limit to how much air you can pack into your thorax. All right, voluntary control. So the motor cortex, remember, that is going to be the prefrontal gyrus, or precentral gyrus, excuse me, and the motor association area. So those are going to send impulses down the corticospinal tracts, familiar from motor control from 231, and those pass along to respiratory neurons in the spinal cord. So these ones bypass the brainstem. There are limitations on voluntary control. So blood CO2 and O2 limits cause automatic respiration. So if you've ever had a kid say that they're going to hold their breath until they get something that they want, go ahead and let them do it. They don't have a choice. At some point, they're going to automatically respire because their nervous system does not permit them to hold their breath any further. All right, chemical regulation of respiration. So I mentioned chemoreceptors in the medulla. So as promised, they respond primarily to changes in proton concentration, but also to the partial pressure of carbon dioxide in the cerebrospinal fluid. So hypercapnia, hyper means above normal, and capnia refers to CO2. So this is an increase in the partial pressure of CO2. And due to a particular chemical reaction that we will discuss later, PCO2 is a little bit more important to monitor than PO2. It seems counterintuitive since the goal of respiration is to get this out of the atmosphere, but PCO2 is a more tightly observed and regulated cue in the body. So there's something called the aortic body in the wall of the aorta, and those nerves join the vagus nerve. There's also carotid bodies in the walls of the common carotid arteries, and those nerves join the glossopharyngeal nerve. So here's just an image of these. The nerves are in yellow. So there's diffuse aortic bodies, which are little clumps of cells throughout the aorta, and there are carotid bodies in the common carotid and the carotid sinus in the internal carotid. Okay, so the effects of hydrogen ions. And I promised I would show you a chemical reaction in a minute here, and I will. So hydrogen ions, H+, which really is just a proton because a hydrogen atom is one proton and one electron. And if you take away that electron, you just get a proton, which is H+. So it's reminding you of some chemistry. That's why I'm going to use those terms interchangeably. Hydrogen cations and protons are the same thing. So the pH of the CSF is the most powerful respiratory stimulus, and that's because you want your brain to be getting enough oxygen. And so you want your brain to notice the potential for losing oxygen first, so something can be done about it, because your brain, of course, is also the control center. So respiratory acidosis is a pH in the blood of below about 7.35 and concomitantly a pH in the CSF of about 7.35. Because remember, where does CSF comes from? It comes from the blood. So this is caused by a failure of pulmonary ventilation. So hypercapnia is PCO2 that's greater than about 43 millimeters of mercury. So here's the deal with CSF. CO2 really easily crosses the blood-brain barrier because the blood-brain barrier, remember, is not able to prevent fatty substances or very small dissolved gases from crossing into it. So it's only really good at keeping out big things. In the CSF, the CO2 reacts with water and releases protons according to the carbonic anhydrase reaction. And central chemoreceptors strongly, I'm going to put the box around this actually, strongly stimulate the inspiratory center. And this is all corrected by hyperventilation. So the hyperventilation in cases of hypercapnia is not actually designed to try to gather more oxygen, but rather to scrub CO2 out of the blood and CSF. So that's what we call it, blowing off CO2. So a proton is combined with this compound, bicarbonate, and that forms carbonic acid. And carbonic acid dissociates into water and carbon dioxide, which you can expire out. The opposite effect is also possible. So respiratory alkalosis, or hypocapnia, is corrected by hypoventilation. So this pushes the chemical reaction to the right. So instead of dumping CO2 by gluing a proton to a bicarbonate, you do the opposite. So you combine carbon dioxide with water to produce carbonic acid. And because carbonic acid is an acid, it donates protons to solutions, producing bicarbonate and a proton, which is a unit of acidity. So the increase in number of protons, it's what's going to push the pH back down to normal. So pH imbalances can have metabolic causes. A primary one that you see a lot clinically is diabetes mellitus. So when you have this kind of diabetes, if it's regulated poorly, all the glucose that comes from the food you eat doesn't enter your cells and tissues and just stays in your blood. So to compensate for this, your body begins to mobilize other fuel sources. So fat and protein get oxidized instead, and fat oxidation causes, ketone bodies to build up in the blood. And this creates a condition called ketoacidosis, which is acidosis of the blood as a result of increased ketone bodies, which donate protons to solutions as well. This can be compensated for by Kussmaul respiration, which is deep but rapid breathing. So in kids with untreated diabetes, you see extreme thirst and polyuria. They can't hold on to water. And you see this deep and rapid breathing, which is a result of the body's attempt to compensate for ketoacidosis. So carbon dioxide has indirect effects through pH, as seen previously, and these are the most powerful. But it also has some direct effects. So increase in CO2 concentration in the plasma may stimulate peripheral chemoreceptors, so ones that are not part of the central set that I showed you earlier, and could increase ventilation more quickly than central chemoreceptors. It's just that central chemoreceptors are better understood and studied. Oxygen usually has little effect, at least in the short term, which is paradoxical because you think of breathing as being designed to collect oxygen, and yet we've just seen that carbon dioxide and indirectly acid have more of an effect on breath than oxygen does. So chronic hypoxemia, which is a partial pressure of oxygen of less than 60 millimeters of mercury, can significantly stimulate ventilation, but it must be chronic, meaning over time, not acute. And emphysema and pneumonia can cause this, so you're just unable to get the gas exchange that you need, either because you've lost lung tissue or because your lung tissue is blocked by a bunch of goop and infection, or high altitudes after several days. So if you decide to go hike Machu Picchu, after a couple days, you may develop chronic hypoxemia, which will be compensated in a variety of ways. So here's just a nice image showing you negative feedback control of breathing, which keeps your breath rate constant as various parameters in your blood or CSF change. So we start with number 1, some disruption of homeostasis. Arterial blood pCO2 or decreasing pH or PO2 is detected by receptors, either peripheral chemoreceptors or central ones. And those are going to start firing off nerve impulses to inspiratory centers in the medulla oblongata. The output from that control center is 2 effectors, which are going to alter your rates of respiration, and then the outcome is you get a decrease in arterial blood pCO2, increase in pH, and an increase in PO2, which brings the whole deal back to normal. So from that, we can infer that the stimulus was less breathing for some reason. So this is just an image showing you the various points at which systemic oxygen transport maxes out for various creatures. So we're not going to get much into this. This is something that Ed likes to talk a lot about, but I'm going to mostly skip in this presentation just in the interest of time. Just note that, so this is the volume of oxygen, so milliliters of oxygen per kilogram per minute, and this is systemic oxygen transport. So they have a proportional relationship, which is pretty tightly hugging the curve line. So an R-squared value means the dots are clustered really tightly around the line, and those vary for different critters. So this is humans, and this is what it looks like for other animals. All right. Regulation of ventilation rate and depth. This is just a figure from not your book, but a different book, showing you and summarizing all of the ventilation rate and depth changes. So they increase, so how fast you breathe and how deeply you're going to inspire, increase with voluntary hyperventilation. So if I'm going to start going really on purpose. Anticipation of activity by stimulation of the limbic system. So let's say you get really, really frightened or angry. That's going to start in the limbic system and then kick off a sympathetic response. An increase in arterial blood protons, or a PCO2 of above 40 millimeters of mercury, and concomitant decrease in arterial blood PO2. So this is detected by chemoreceptors. Increases in sensory impulses from proprioreceptors in muscles and joints will also do this, and that's because if you're propriorecepting more, that means you're moving around more, and you'll probably need to supply those active muscles with oxygen at higher rates. A decrease in blood pressure also changes your breath rate, as does increase in body temperature, prolonged pain, or stretching of the anal sphincter, oddly enough. Ventilation rate and depth decrease with voluntary hypoventilation, but that's limited, of course, by the buildup of protons and carbon dioxide. So you can hypoventilate or hold your breath only so long before your brain will force you to inhale. A decrease in arterial blood proton levels. So, respiratory alkalosis, and a decrease in arterial blood PO2. Decrease in sensory impulses from the proprioceptors. So let's say you're laying down, you're going to take a nap, your ventilation rate and depth will decrease. And increase in blood pressure will also do this, as will decrease in body temperature. Severe pain can cause apnea, so if something hurts so bad, you might momentarily stop breathing. And irritation of pharynx or larynx by touch or by chemicals. And that causes apnea followed by coughing or sneezing. Okay, so I've been using the term PCO2 and PO2. We need to talk a little bit more about what that means. So with regard to the composition of air, air is a mixture. So the air we breathe every day is not only oxygen, it also contains nitrogen, hydrogen, and all of these come in the form of molecular gases. So N2O2, H2O is water vapor, and then CO2. Most of the air we breathe is nitrogen, so it's 78.6% of the atmosphere. So if you multiply 78.6%, which is the proportion of atmospheric gas that is nitrogen, times 760 millimeters of mercury, which is 1 atmosphere of pressure, you get the partial pressure of nitrogen. And that's molecular nitrogen, not nitrogen atoms. And you can do the same with the other gases. So partial pressure of O2 is about 159. Partial pressure of water vapor is 3.7. Partial pressure of CO2 is 0.3 millimeters of mercury and rising, because as we know, CO2 is building up in our atmosphere due to human activity. So partial pressures determine the rate of diffusion and gas exchange between the blood and alveolus. So the driving force for gas exchange across the respiratory membrane is the difference in partial pressures between your blood and the alveolar air. So speaking of alveolar air, here are some differences. Outside air is about 21% oxygen, 79-ish%, these are abouts, not precise figures, and about 0.3 to 0.4% CO2. By the time the air gets to the alveolus, the O2 percentage is about 14, 79% N2, and 5% CO2. And that's because, of course, carbon dioxide is diffusing from the blood into the alveolar air. And the air you breathe out is about 16% oxygen, 79% N2, and 4.5% CO2. So notice that nitrogen concentrations remain the same. Why? Well, there's no difference and there's no driving force there. Alveolar air has less oxygen since the oxygen is being absorbed by the blood. Mysteriously, expired air has more oxygen and less carbon dioxide than alveolar air. Weird. And remember, the anatomical dead space is the 150 milliliters of the 500 milliliters of tidal volume that doesn't participate in gas exchange. So some of the reason for that mystery is that on the way back out, alveolar air is mixing with air that never did gas exchange, which changes those numbers. All right, the air-water interface, and this brings us back to some of those laws. So Henry's law states that the amount of gas that dissolves in water is determined by its solubility, but also by its partial pressure in air, which is a big deal, as we just saw. But generally speaking, gases diffuse down their concentration gradients. So if I have a membrane, and let's say that red dots, let's say that red dots equals oxygen. In this picture, if this is my membrane, and I have more red dots over here and fewer down here, the red dots are going to go this way, right? So let's see what happens if I have two opposing gradients. Now let's say that blue dots equal carbon dioxide. The blue dots are going to go from where they're most concentrated to less. So even though you might even have equal numbers of gas molecules in total on each side, because of the driving force of partial pressures, this is going to diffuse this way and this is going to diffuse this way. All right, so Henry's law. The quantity of gas that will dissolve in a liquid depends upon the amount of gas and its solubility coefficient. So this is why you can breathe compressed air while scuba diving, even though it's 79% nitrogen. Here's why. So nitrogen has a very low solubility in water, unlike CO2. When you dive really deep, increased pressure from the water forces more nitrogen to dissolve into the blood than normal. So you can change the solubility of a gas by changing pressure. You can force more gas into it. So this is called nitrogen narcosis, which is a feeling of sort of brain fog and malaise when you're scuba diving very deep down. Decompression sickness can happen if you come back to the surface too fast or if you stay deep for too long. So if you are scuba diving very deep, and I know because I've done this, you have to, on your way back up, you have to stop in the water column and wait, typically for between 10 and 15 minutes, sometimes longer. And that waiting that you're doing is waiting for the decrease in pressure to allow some of that nitrogen to diffuse out of your blood via your breath. If you don't do that, if you shoot back up to the surface, the nitrogen will form small bubbles in your blood, and that gives you the bends, which can kill you because an embolus of air in your bloodstream is not very good. All right, so hyperbaric oxygenation. So hyperbaric means high or above, and then pressure. So let's say you do get the bends, or you have any number of other problems. You might be put into a hyperbaric chamber, and this uses pressure to dissolve more oxygen into the blood than normal, or to reverse the effects of nitrogen narcosis and embolus. So it's a good treatment for patients with anaerobic bacterial infections. So remember, anaerobes, especially obligate anaerobes, they hate oxygen. They're killed by it. So if you force more oxygen into the blood, you can kill anaerobes. So if you have tetanus and gangrene, you can get stuck in one of these chambers, and that will help the anaerobic bacteria to die. Hyperbaric chamber pressure is raised to 3 to 4 atmospheres, so that's 3 or 4 times the normal pressure, so that tissues absorb more oxygen. It can also be used to treat heart disorders, carbon monoxide poisoning, because carbon monoxide competes with oxygen for a spot on your hemoglobin molecules, cerebral edema, bone infections, gas embolisms, so this is the scuba diving accident, and crush injuries. So with alveolar gas exchange, during oxygen loading, you've got the initial state here and the equilibrium state. So oxygen in the form of blue balls here is going to move into the blood until they reach equilibrium. Because remember, everything still obeys the laws of diffusion. So once equilibrium is reached at a given pressure, no more exchange occurs. CO2 unloading, in the initial state, we've got lots of CO2 in the blood and not much in the air. And then equilibrium is reached again. So at the alveolus, the time required for gases to equilibrate is about 0.25 seconds, so not very long. Red blood cell transit time at rest, so how fast it gets through the alveolar capillaries at rest, is about 0.75 seconds to pass. Red blood cell transit time with vigorous exercise is about 0.3, so it never dips below this 0.25, which gives you enough time for gas exchange, which is good. So factors affecting the exchange of gases include, of course, solubility and concentration, but also other parameters. So let's talk about those. The partial pressure of oxygen is about 104 in alveolar air versus only 40 in the blood. So that's a very, very strong driving force. The difference between partial pressures of CO2 in the blood and in alveolar air is much smaller, but still provides a driving force. So just less of an arrow. So let's make this a big, thick arrow. And this is a modest arrow. Carbon dioxide, however, is 20 times more soluble. So oxygen has a bigger concentration gradient. Carbon dioxide has larger solubility. So remember, the membrane thickness is only about 0.5 microns thick. And the membrane surface area, if you spread it all out, is over 70 meters squared, which is a lot. Ventilation perfusion coupling is areas of good ventilation need good perfusion, and the opposite is true as well. So if you are, let's say, breathing quietly and passively and not inflating all of your lungs, why would you send blood to dead areas of your lungs to get oxygen? You wouldn't. So that's what ventilation perfusion coupling is for. So this is just a picture showing you the relative partial pressures of oxygen and CO2 respectively throughout various points in the body. So oxygenated blood, I'm going to put quotes around this, has a PCO2 of 40 and a PO2 of 95. Quote-unquote deoxygenated blood still has a PO2 of 40 and a PCO2 of slightly more at 46. So even though deoxygenated blood is called deoxygenated, we don't get all the oxygen out of our blood in one circulatory go. That's not how it works. So there's 40 millimeters of mercury worth of oxygen left, and that's what we call our venous reserve. At the tissue fluid, the PO2 is about 40 millimeters of mercury, and the PCO2 is 46. So oxygenated blood interacting with the tissue fluid is going to tend to give up oxygen and receive CO2. Ambient pressure does affect concentration gradients. So air at sea level, or 1 atmosphere, we have a normal gradient and normal O2 diffusion. So this is concentration gradient of oxygen. Here we have venous blood arriving at the alveoli. So the ambient PO2 is going to decrease here. So the air in a hyperbaric chamber is really pressurizing the air and therefore the CO2, so you have a super steep gradient. And the lower you get with ambient O2, the reduced gradient creates slower diffusion, which produces chronic hypoxia and hypoxemia. So let's discuss lung disease a little bit. So here we have emphysema. Now let's count alveoli. And of course, I'm not going to exhaustively do this, but 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28, 29, 30, 31, 32, 33, 34. So I count about 35. 35. Let's count them here. 1, 2, 3, 4, 5, 6, 7, 8, 9, about 9. So this difference in surface area is significant. So the less surface area you have, the less gas exchange you can perform with the alveolar capillaries. So confluent alveoli or having two alveoli merge is bad. So this is what emphysema does to you. It causes you to lose lung tissue. Pneumonia causes a thickening of the walls due to edema and fluid buildup. So increasing membrane thickness or decreasing surface area are both deleterious to your ability to exchange gases at the alveolus. Okay, so let's look at ventilation perfusion coupling. So let's say you have an increase in airflow. you're going to have an elevated partial pressure of oxygen in the blood vessels, and you will have vasodilation of the pulmonary vessels. That's going to increase blood flow so that blood flow matches airflow. So you're going to have blood flowing to where air is in this circle. If you have decreased airflow, why would you send blood to a place where there is no air? So you have reduced partial pressure of oxygen in those vessels. The pulmonary vessels in those oxygen-poor places vasoconstrict, resulting in decreased blood flow, such that, again, blood flow matches airflow. And ventilation adjusts to changes in perfusion as well. And so we, of course, know that this is partially due to your pneumotaxic and apneustic centers, but also due to the following. Let's say you have decreased blood flow to an alveolus. That means you're going to have reduced PCO2 in the alveolus, constriction of the bronchioles, and decreased airflow. Increased blood flow means you're going to have elevated PCO2 in the alveoli, because you're dumping CO2 out, meaning dilation of bronchioles and increased airflow. So in this way, airflow matches blood flow as well. So now we can separate external and internal respiration. So external respiration is exchange of gases with the atmospheric air. So with external respiration, deoxygenated blood becomes saturated with oxygen as much as it can be in the time and solubility given. So you get PO2 of 159 dropping to PO2 of 105, and then once we get to oxygenated blood right by the heart, PO2 is about 105, and then it drops down to between 95 and 100 by the time you get to a systemic capillary. The rate of diffusion depends on the partial pressures of gases in air. So the PO2 at sea level is about 160 millimeters of mercury. At 10,000 feet, it's only 110 millimeters of mercury, and at 50,000 feet, it's only 18 millimeters of mercury. So if you are a fighter jet pilot and you are going to spend quite a bit of time way super high up in the air, wearing a pressurized suit is going to be one of the things that keeps you alive, partially because it keeps the concentration gradient of oxygen high enough for you to survive. So, other things that influence the surface area, or the diffusion of gases, are surface area. So I showed you with the emphysema slide how losing surface area decreases gas exchange. The diffusion distance is really small, so only 0.5 microns. And the solubility and molecular weight of the gases. So O2 is smaller and therefore diffuses faster because it can wiggle through membranes easier. CO2 is bigger, but it dissolves 24 times more easily in water, so the net outwards diffusion of CO2 is much faster. Disease produces hypoxia before hypercapnia, which is a lack of O2 before too much CO2. All right, so the reactions in alveolar gas exchange are the reverse of systemic reactions. So CO2 unloading states that as hemoglobin loads O2, so O2 is leaving the alveolar space, going into the blood and binding to hemoglobin, hemoglobin's affinity for protons decreases. So up until now, oxygen-poor blood, the hemoglobin in it has been holding on to protons, but loading of oxygen reduces that, so hemoglobin starts kicking off protons. So those protons dissociate from hemoglobin and they go and bind to the nearest bicarbonate. That produces carbonic acid. And then you get water vapor and carbon dioxide vapor exiting from the blood into the alveolus. Now, let's talk about the chloride shift, which is also called the hamburger shift, which is hilarious. It's because the guy's name was Dr. Hamburger. So, bicarbonate diffuses back into the red blood cell in exchange for chloride. Why? Well, you need the bicarbonate inside the red blood cell in order for this reaction to occur. So, in order to do that and still have the same osmolarity and charge separation across the red blood cell membrane, you have to exchange one anion, this one, for another. So you don't get a net anion loss by giving up that bicarbonate, you're just trading it for a chloride. So free CO2 is generated and diffuses into the alveolus to be exhaled. And that is where I'm going to stop for today, so stay tuned for the conclusion of the respiratory lecture in the next screencast, which is going to be the respiratory system part 4, the final chapter, or at least I hope. Thanks for your attention, and I'll see you next time.